So at this time, it is my honor to introduce our final portion of the meeting, which is our IAQ Area of Interest presenters. Today, I want to remind people that you can put any questions into the chat box at any time during the presentations. And we have allowed time after each of the next two speakers for questions and answers. So today we have two speakers, both coming out to us from Lawrence Berkeley National Laboratory. And we're going to begin with Dr. Jacob Bueno de Mesquita, and I am sure I butchered your name. I am really sorry, so please say it when you get ready to speak. Jacob is in environmental health epidemiology. He dedicated much of his doctoral and postdoctoral study to quantifying modes of influenza transmission, characterizing viral shredding, including the SARS-CoV-2 virus, in exhaled breath aerosols, and identifying contagious biomarkers. Today, he will be speaking to us on the topic of how buildings can stop the spread of SARS-CoV-2, evidence and research priorities. Jacob?
Hello, thank you very much for the introduction. My name is Jacob Bueno de Mesquita. I'm a postdoc at Lawrence Berkeley Lab. And for the last year or so during the pandemic, we've been hearing about how SARS-2 has been being transmitted in poorly ventilated indoor environments. And this presentation is going to focus on the control of airborne infectious disease in buildings and the evidence that we have for these controls and research priorities to build new evidence base. The question we're asking here is, how can buildings prevent aerosol transmission? We wrote a review paper on this topic, which should be published shortly. And in that paper, we did a simple model of exposure to exhaled breath aerosols at different scales in the indoor environment, up close, across a room, and between rooms. We also reviewed the literature of engineering control effectiveness, and from that, we identified key knowledge gaps and research priorities to improve this knowledge base, ultimately to help make indoor environments safer and more pandemic resilient. I wanted to start by acknowledging that infectious SARS-2 virus has been detected in exhaled breath aerosols. This is work from the team at the University of Maryland. They recruited SARS-2 cases, COVID cases from the campus community and local College Park community in Maryland. These people were not very symptomatic, and most of them were recruited early in their illness, generally between day 2 and 5 post-symptom onset. A couple of the people in this study were not symptomatic at all. They were asked to come and give a sample in the Gesundheit 2 machine, which is what's shown here. Oh, sorry, I didn't indicate to advance slide. We should be on slide 3 now. Hello?
Yep,
thanks.
it's on slide 3. Yep.
Okay. So in the figure on the top left, this is the Gesundheit 2 machine. It collects exhaled breath aerosols into what we call fine aerosols, which are those less than 5 microns, less than or equal to 5 microns, and coarse aerosols between 5 and 80 microns. And we asked people to sit in the Gesundheit 2 machine for 2 half-hour sessions, one with a mask and one without a mask. So we could also measure the effectiveness of the mask on the aerosol shedding. And what we found was that virus was detected in 45% of the fine aerosol samples, 31% of the coarse aerosol samples, and 65% of fomite samples. Fomites were people's mobile phones. And there were 57 people in the study, and 4 of them were alpha variant infections. And so it's a very small sample size, but we did see that those people with alpha variant in the study were shedding 18 times more virus into the fine aerosol fraction after controlling for the viral load in the mid-turbinate swab and cough and other covariates. We also found that masks captured 48% of the virus in the fine aerosol fraction and 73% of the virus in the coarse aerosol fraction. If you look at the figure on the top right, you can see that the first 3 violin plots on the left correspond to MTS, mid-turbinate swab sample, saliva sample, and fomite sample. And the y-axis here is the viral load in log 10 RNA copies. And so you can see that we're seeing almost up to 10 to the 10 RNA copies in the mid-turbinate swabs at the maximum, a little bit less for saliva, and a little bit less still for the fomite, although we're still seeing a range up to 10 to the 6 RNA copies. And then the rightmost 4 violin plots are showing the aerosols, the coarse aerosol greater than 5 micron without the mask in blue and with the mask in red, and the fine aerosol as well, without mask in blue and with mask in red. And I'm circling this last plot in red, the fine aerosol plus the mask, because this was where 2 fine aerosol samples where people were wearing masks were found to be culturable. So infectious virus was detected in those samples. And this is despite there being relatively lower viral load here. And generally, it takes maybe 10 to the 5th or 10 to the 6th RNA copies for there to be detection of culturable virus in these sorts of lab assays. But there were still 2 samples that were culture positive there. This does not mean that most of the fine or coarse aerosol is non-infectious. Probably the receptor cells in actual human respiratory tracts are more sensitive to infection than the varocell cultures used in studies like these. But this is some of the first evidence that's actually cultured virus from exhaled breath. Next slide, please. So we wanted to do a model of aerosol transfer between individuals, an infectious person and susceptible people in a room. And in thinking about setting up this model, we thought that it would be helpful to set up different scales of exposure, and based on how far away people are from each other. But so we have this close interactive scale of exposure where people are within a meter or 2 of each other. You may think of this as like the 6-foot rule that has been widely talked about. And about 15 minutes of exposure time within a day. And we also thought it would be helpful to think about this in terms of people that are conversing, eating, or laughing together, because that's what often happens during close interactions. So you can see this illustrated in the figure on the right with the figure circled in orange standing next to the infector with the red aerosol plume coming from them. Then there's the room scale exposure. Within room, non-close interactive exposure. And this is talking about the other people, the other susceptible people in the room that aren't right next to the infector. And this is where there's a large potential to have a large number of exposed individuals, such as in a restaurant or in a performance hall or in a classroom, where there could be many people exposed. And also in poorly ventilated environments, the viral aerosol can build up in those room settings. So last, there's the building scale of exposure, and here we're talking about exposure between rooms and floors, and that's illustrated with the blue circle, the person sitting in the room next door to where the infector is. And this is mediated by aerosol transfer through forced air HVAC systems, doorways, halls, plumbing, common exhaust fan inlets. Next slide, please. So in thinking about modeling approaches for aerosol transmission, there are several different approaches that have been used. The Wells-Riley approach is still very, very commonly used. And this approach is drawing a relationship between infection risk and exposure. And it assumes a well-mixed environment. And the main factor that's mediating exposure here is the ventilation rate in the room, as well as how much virus the infectious person is shedding, the infectious dose generation rate, and how much volume of air susceptible people are inhaling in the space. This Wells-Riley model has been iterated on with the rebreathed air equation that uses CO2 as a main indicator of exposure to exhaled breath, because humans exhale a lot of CO2 in their exhaled breath compared to outdoor levels. And so it's a good indicator of how much air people are breathing in contains exhaled breath from others in the room. It's also dependent upon occupancy because the number of people in the room is directly related to how much exhaled breath is there. So this model is nice for estimating exposure to the very fine particle aerosols that are thought to be important for transmission, those less than 5 microns that I was talking about earlier. And there's a lot of nice public health applications because, as was mentioned earlier, people can use CO2 sensors and take them with them into indoor environments and get a sense for what their risk might be for airborne transmission should there be an infectious person in that space. But we are also interested in understanding the potential contribution of particles both below 5 microns as well as perhaps just above 5 microns, up to 10 or a little bit higher. And so we considered a CONTAM model approach that CONTAM, which was sort of mentioned by Lisa Ng earlier at NIST. And this approach is considering different particle sizes. And it's not making any assumptions about the risk of transmission associated with the exposure. But this is putting putting a model in terms of where we can talk about engineering controls with 2 different particle sizes that might be involved in transmission. And for SARS-2, probably the fine aerosols are important, maybe some aerosols just over 5. But for different infectious diseases, the distribution of where the infectious aerosols are could vary. And so having a flexible model could be helpful for this. Next slide, please. So we looked at setting up a model to evaluate exposure in an office for 1 hour. This model assumed exhaled breath particle distribution previously published from studies from Lydia Murwaska's team. We assumed that particles shrink by 40% due to evaporation, and that in the room there was 7.5 liters per second per person ventilation, corresponding to 10% outdoor air coming through the HVAC system. We also assumed there were MERV 13 filters in the HVAC system. If you look at the figure on the top right, you'll see that there are 2 people in this little pink box. The person in red is the infector, and the person in black is the susceptible person. And this is the subzone. It's simulating the close interactive scale that I mentioned earlier. That's that pink, where the pink dot is. So it could be a little challenging to do something like this in person, trying to stick people in real life in a box. So we have this crafty model to do it, to sort of simulate this up-close exposure. And then the blue dot here is representing the room scale transmission within this room of 93 meters squared space, or 1,000 square foot area. And then down below, we have sort of the same setup, but we've added another room that's served by the same HVAC system in an office environment, and that green dot is representing the between room or building scale transmission. And also this brown dot here is referring to the room scale transmission, but that exposure changes a little bit when you add HVAC that's connected to other rooms. So we wanted to account for that in the model. Okay, next slide, please. So in running this little simulation, we found that the exposure scales, masks, and particle sizes all affect exposure. They're all important in affecting exposure at these different scales. So here in this figure, at the very top, I should just let me explain the axes. So the x-axis is showing on the bottom the initial particle size, and we're looking at particle sizes submicron all the way up to 40 micron, and then those particles are shrinking by 40%, leading to the equilibrium particle size at the top of the top of the chart here, and the y-axis is talking about the ratio of inhalation exposure in the close interactive scale compared to the other scales. So the reference group is the close interactive scale. And so that is showing up as 1 on the y-axis. Those peach-colored open circles are 1, and the open circles are representing the unmasked scenario, and the closed circles are representing the masked scenario. So the masks are leading to overall a 53 to 98% reduction in exposure across these different scales, which is what we would expect given that we put a mask on that had that sort of level of capture efficiency. The room scale is in blue here, brown, and also blue. The brown is the single zone where we weren't considering an adjacent room served by the same HVAC, and the blue is the multi-zone where we also included the HVAC attached, serving multiple spaces. And you can see that the exposure level compared to the close interactive scale is pretty similar at room scale for both of those scenarios, although you get a little bit less exposure when in the scenario where the HVAC system is connected to other spaces, because you're getting a little more air slow dilution ventilation coming from that. And then the green is looking at the between room or building scale aerosol transfer. And you can see that it is dramatically lower than the other scales. So this is showing that the risk of aerosol transfer between rooms is quite low. We also see that the rate of exposure reduction is increasing with the larger particles. So as the particle sizes are getting larger, they're depositing on the surface more readily, or being captured by mask or filter more readily, and not posing an inhalation exposure risk. Okay, next slide, please. So we also showed that exposure to aerosol volume can increase with particle size. Here, this time on the y-axis, we have the volume, the amount of exposure to particle by volume, not by particle number, but particle volume. And so a larger particle could contain a lot more virus if you assume a even distribution of virus across particle size. And so a single larger particle at 10 microns, for instance, as you can see in the top right corner here in peach color for a close contact interaction, is sort of the reference group. And the lower, on the y-axis going down, you can see that the exposure is decreasing in terms of volume of aerosol inhaled by people at these different scales as the particle size is decreasing. So this brings up the question about how many infectious particles are there per aerosol particle at these different sizes, and whether it's really an even distribution. But we wanted to show the impact of the volume here as well as the number of particles being exposed. Next slide, please. So we then did a literature review of these different control measures or strategies that we have, engineering control strategies for buildings. And I'm just going to share a few of the highlights from room scale control measures. So this is talking about ventilation at the room scale. And for hospital settings, they generally have stringent standards for ventilation, but it's for infection control specifically. But outside of the hospital setting, ventilation standards are not generally set for infection control, but rather for occupant satisfaction, thermal comfort, and schools commonly actually have below the standard set. During the SARS-2 pandemic, we're seeing a lot of case studies that are linking ventilation with SARS-2 transmission, such as those in a restaurant or a call center or a bus. For example, this one study in a restaurant showed that they would have needed 38.6 liter per second per person ventilation to have avoided transmission in the restaurant, but the restaurant only had 0.9 liter per second per person, and transmission was observed there. So a bunch of epidemiologic studies have shown increases in transmission risk for respiratory infections, influenza and other respiratory infections, by 25 to over 300% in the lower versus higher ventilated spaces overall. This on the right lower figure is showing some buildings from a study published at University of Maryland campus that looked at a low ventilated environment and a high ventilated building environment, and found some differences in infection rate over the course of a semester by prospectively measuring infection in individuals living in these dorms. We also know that windows can achieve abundant ventilation, but they are perhaps not reliable for providing steady source of ventilation. There's also issues with temperature regulation, safety, and so forth. Next slide, please. So next is filtration for room scale control. So there is the HVAC filtration and the standalone air cleaners pictured here on the top right. Lots of studies have shown that air cleaners work well. We know that theoretically they remove particles efficiently from the air. People have been adopting this do-it-yourself box fan filter for SARS-2, shown here on the bottom right. And these are great. It would be nice to have demonstration studies to actually show the benefit of adding these filtration systems on actual infection rates in schools. Next slide, please. So next is germicidal UV for room scale control. This is a 100-year-old technology that has been proven to be really effective. Upper room GUV with ceiling fans was associated with tens to hundreds of equivalent air change per hour in experimental chambers, and it's been shown effective epidemiologically in a VA hospital for influenza and in schools, K-12 schools for measles outbreaks in the 1930s and 40s. On the lower right is a picture. In this black and white picture with the students reading, there's a little kind of round unit in the ceiling, and that's the GUV unit that's shining upwards towards the ceiling and irradiating the air. And this was associated with reducing the severity of measles outbreaks in these classrooms. And you can see in the chart on the bottom right, and primary classes irradiated in this Germantown school in the 1930s and 40s, measles rates were substantially lower despite similar number of introductions into the school compared with upper classes in the same school that were not irradiated. And this is despite that in the upper classrooms, the upper grade levels, the susceptibility to measles is much lower because many people have already had measles by that age. So FAR GUV is being developed and tested now, but early results are showing that it's also safe and effective. FAR UV uses a lower wavelength that is not able to penetrate the eye or the skin, and so could be shined directly into the room. Overall, the big benefit of GUV is that you can irradiate and sanitize larger volumes of air, much larger volumes of air than you can by drawing air through a filtration system or an HVAC unit. There's also some benefit by putting it in duct to reduce biofilm and molds. Next slide, please. So Haran is going to talk some about this in the next presentation, but airflow management can also be used for room scale control. Some studies have shown that displacement ventilation is reducing exposure within the room, and also that thermal strata can reduce the rate of dilution in the room. And in the lower right here is a picture from a study done at the Flux Lab here at Lawrence Berkeley Lab, which Haran is going to talk more about. But managing airflow is, and being able to achieve well-mixed environments and not create hot spots for exposure within a room is something that is being worked on. Next slide. So we wanted to note that building scale transfer can happen even though our model is showing that the likelihood of it, of building scale exposure is much lower than at up close and within room scales. So SARS, MERS, and flu superspreading have been observed within hospital settings, and SARS superspreading has been observed through a high-rise apartment building between units. And also dorm room airflow connections has been associated with probable influenza transmission. And this is what's shown on the right-hand slide. These little cubes are dorm rooms, and this double room, room number 3 with the asterisk on it, in the top row was where a primary case was. And it was actually shown through a little air model simulation that this room down below in the bottom right, room 5, was the only room connected to room 3 by airflow, by an airflow connection, just based on the way the air was moving in the building. And so there could have been a transmission between room 3 and room 5 here. They each had positive cases for cold within a few days of each other. Next slide. So that was just a brief summary of some literature that about using buildings, how buildings might either contribute or be used to mitigate risk. But through that, we identified some knowledge gaps. One being we need more information about infection pathogen shedding rate into fine and coarse aerosols across the aerosol size range, and susceptible sites in the respiratory tract, and also the infectious dose. We also want to learn more about how to reduce close interactive exposure with engineering controls, since that seems to be where the risk is highest, although across room risk can affect more people. Which settings have the greatest opportunity for equitable intervention is another important question, and also how to improve models to predict risk. How to gain greatest infection prevention from controls in various settings, how to use and combine controls for most cumulative health benefit with the least energy cost, and how to implement effective controls in buildings at scale. Next slide, please. So from these research gaps, we identified research priorities for well-characterized environments, as well as practice-based environments in the field, and also research priorities for methodology. So in terms of well-characterized environments, we found that it would be important to measure size-resolved aerosol transfer in the highest risk settings. And we're talking well-characterized environments here, either chamber studies or small-scale real-world environments that can be very well-characterized. Number 2 is to characterize boundaries between close interactive and room-scale exposure under different control regimes. This is asking, is the 6-foot distance an important boundary between close interactive and room scale exposure, and how does that change under different types of controls? If we can achieve room scale type exposure at 3 feet, that would be even better than 6 foot. But so that's something also to explore. The next is to use computational fluid dynamics to support simple within room spatiotemporal aerosol evolution models, and 4, to evaluate germicidal UV and air cleaner effectiveness with infectious surrogates when deployed in high-risk settings. and also of lower priority to quantify potential risk from resuspension. Next slide, please. So the research priorities for practice-based settings, we identified 5 of them. One was to evaluate, control, implementation, and support outbreak investigations, such as those ongoing for SARS-2, but also to invest in population-based epidemiologic studies of control effectiveness. What we have now is a lot of case reports about how different controls might work or not work, or how poorly ventilated spaces are associated with increased risk, but we really need larger scale epidemiology to better demonstrate the effectiveness of various controls in different settings. 3, to conduct comprehensive aerosol infection risk assessment for indoor environments. This risk assessment piece being really important. And number 4, to identify high priority settings for controls based on building operation, use, and climate, to identify really where we could get the most benefit by focusing implementation of controls. And 5, to identify common challenges to implementation, such as operational challenges, energy or cost challenges that might be hindering the implementation of engineering controls in buildings. Next slide, please. So lastly, we identified research priorities for methodology. There were 4 of these. 1, to develop metrics and test methods for infectious aerosol inactivation. It would be nice to have something similar to a clean air delivery rate for a GUV system. So if a school is asking, what kind of benefit can I get from putting a GUV fixture in a classroom or a dining room, it might be a little easier to get a general sense for the benefit that could come with that. Although there is benefit, it'd be helpful to know sort of more exactly how much benefit. 2 is to develop methods to improve sensitivity of infectious aerosol detection in the air. And there are some teams that are working on this already. Detecting infectious aerosol from environmental samples is often challenging because it's so dilute, and then getting it to actually grow in cell culture is often another challenge. 3 is to prepare models that predict energy use and infection risk, implications of controls. Lisa Ng mentioned earlier that there was a way to sort of link Contam and Energy Plus. And that's sort of one way of exploring this sort of methodological area of research priority. Number 4 is to develop standardized methods for investigating risk of infectious aerosol exposure. And this would be really helpful since people across the country, across the globe are definitely recently with SARS-2 investigating risk of infectious aerosol exposure. It would be helpful to have some standardized methods. And this can even help later when trying to link across studies to do meta-analyses and so forth. Next slide, please. So overall, there are sort of 3 categories of overall research needs, although I sort of described a lot there. So 1 is more demonstration of performance of control measures in a variety of different settings. 2 is to identify and address barriers to widespread deployment. And 3 is to evaluate the effects on health beyond infection control. And this could be useful for really understanding the benefits that we can get by improving the indoor environmental quality of different spaces. Next slide. And that is all. So I'm happy to take any questions. Thank you.
Thank you so much for that presentation. We did have a couple of questions for you. Just reminding everyone that if you have any questions, please use the questions function to submit them. The first is, when testing for the coronavirus in the experiment, was there a difference in aerosol characteristics for the different variants?
There was more virus. So in that study, you're talking about the University of Maryland study. There were only alpha variants and kind of wild type variants, we're calling it, sort of before alpha emerged, included in that study. And so for the alpha variants, we did find that they were shedding more virus into both the coarse and the fine aerosols, even while controlling for increases in viral load in the upper respiratory tract, measured by mid-turbinate swabs and saliva samples. There was no, one of the samples, one of the fine aerosol samples that was culture positive was an alpha variant, and one was not. But that's all the information available about the different variants. There were no delta variants in that study, as it hadn't arrived in that area at that point.
Thank you. Another question, what kind of masks were used during the University of Maryland study?
Whatever mask the study participant wanted to wear was what was used. And so generally it was either their own cloth mask or a surgical mask that they brought or we provided. Occasionally people were bringing an N95 that was not fit tested, but it was mostly cloth masks and surgical masks.
Thank you.
So the study is giving a sense of a general mask that might be used in the public at a population level.
Great, thanks. How long were the virus particles in the air?
Is this referring, I'm not sure what's being referred to here.
So the clarification was the University of Maryland study. I guess how long were the virus particles in the air in the study?
So the virus is being sampled directly from the exhaled breath of the people that were giving sample in the Gesundheit 2 machine. So it was not in the air for very long, for, on the order of, less than a second, being sucked into the Gesundheit machine.
Thank you. What indoor air quality technologies or mechanisms have shown to be the most effective in your experiments?
Well, I think this is sort of one of the areas of research priority. In the review of the literature, we see that ventilation and filtration and GUV are all can all achieve a great level of control in different indoor settings. Germicidal UV can achieve perhaps the largest air change per hour equivalence, just because it can sanitize such large volumes of air per unit of time. So various studies have shown that you can achieve in the range of hundreds of equivalent air changes per hour. Generally, these are when you have abundant GUV in the upper room and you're circulating the air, you're drawing the air into the upper room with a ceiling fan. So you're getting a lot of air movement between the sanitization zone in the upper room and the breathing zone in the occupied space below.
Thank you. Let me see if we have any more questions coming in. All right, I think that's it. Back to you, Lorraine, to introduce the next speaker.
Okay. Sorry about that. My phone wouldn't unmute there for a minute. Thank you so much. That is so interesting, and it will be very interesting to see where this all goes, not just for COVID, but, for other viral and other kinds of infections that are spread indoors. And it'll be interesting to see what we can learn as we move back into buildings full time.